Yeah, it's my pleasure to give you our year two update for the FaceBase project. This obviously reflects the work of many members of the team here, as well as contributions of the community too. Um, Next slide, please. All right, there we go. Okay. So just as a little snapshot of where we stand here in FaceBase, we now have over 1,132 data sets. And the thing to keep in mind about a data set is that, in some contexts, a data set means a file or a spreadsheet. In FaceBase, a data set is actually a collection of a lot of data. And so a single data set could be what others refer to as a database of sorts, could be thousands of subjects or hundreds of specimens in it. So it's really substantial collections of data here. We now have over 100,000 image files alone. We have many more other types of data, as I'll show later in the presentation. Robust usage continues. A lot of usage in the United States, of course, but a great amount of international usage as well. So the resource is used worldwide. 18,000 annual visitors. And if you can see the number here, so data downloads this past year of 94,000 is really a spectacular number. I like to kind of remind our audience that 10 or so years ago, if we had about 1,000 file downloads, that was a great year, and just generally celebrated. Now we're looking at tens of thousands of downloads in a year. So really robust usage of face-based data. I will just say that our mission continues to be to provide really a premier data resource for dental, oral, craniofacial, inner ear related biological resources. Our aims are to provide a comprehensive data resource in support of advancing research, to promote multidisciplinary collaborations throughout the community, to integrate genomic and phenotypic and all related data, and then to enhance data reproducibility, especially now with AI and ML readiness being in focus. Just a reminder or an introduction to anyone new, FaceBase is really built on fair data principles, and these lead right into AI/ML ready data. So what that looks like in practice is that you begin by finding data on FaceBase. This is enabled by the careful categorizing and annotating of data into a rich model for it. Then being able to access that data, and we have a lot of not only high-level details, titles, descriptions, high-level keywords, but all the data is then structured into experiments and biosamples, and you can tell exactly how files are related into that structure and how they relate to each other. And then finally, it's interoperable because of controlled vocabulary and reusable both on the site. We have a number of different visualization capabilities, and of course, you can download the data through a reliable archive mechanism. So all of that is built on a technical platform here that we have built up, and other groups within our work here at USC in an open source platform called Deriva. And this has all sorts of tools that enable what you see in FaceBase. So online database services, object storage services, interfaces that adapt to the data model as it changes. And so the core of that is data management. So Deriva is a fair data platform. We support multimodal data across genomics, imaging, and really any kinds of scientific data. We have both controlled access as well as open data, and all of this benefits from strides, and in strides we do intelligent tiering to keep our storage costs down. On top of that, we layer capabilities of visualization and discovery. So as I mentioned, we have various 3D viewers, and we integrate with the UCSC Genome Browser. We're a public hub for that. We use the UCSC Cell Browser and other online viewers in our system. And then finally, of course, we're pushing into AI and just other areas of interoperability. So I'll be talking more about that later. But face-based data is AI-ready data, and we'll be presenting some new capabilities around that. So as I mentioned, the FaceBase data is a very broad collection of data, and we might be best known in some cases for imaging data, the tomography and MR data sets, but really there's gene expression, epigenomics, there's even material analysis data in FaceBase. Well, back to imaging, there's large collections of facial scans, and as I mentioned, some of these are data set counts, so it's kind of apples to oranges. I mean, some of these data sets may be really huge. Facial scans databases have thousands of human subjects worth of facial scans in them. So don't let the bars there fool you. There's really large amounts of data all across these diverse collections. Just to take a snapshot of what the pipeline looked like in this past year, we received, there were 49 new data submissions. I think that's a very robust year, and 17 new projects onboarded. So I think that's a testament to keeping our processes kind of streamlined, because if you recall in the past, we had about 10 dedicated projects for a 5-year period. Now we have to onboard almost twice that many in a single year. And so we've had a really robust year of data submissions. We have about 15 new data sets that are released, and then there's another almost 70 data sets that are either currently being curated, or 35 of those have already been fully curated and are just pending publication of their accompanying manuscripts before they're released. So we do embargo data sets. So you can contribute data to FaceBase. We'll keep it secure until you're ready to have it publicly released. I don't have a timer, so if you can let me know when I'm at my time, or a few minutes before. Thank you. Of course, a big part of our focus is really expanding and supporting the entire research lifecycle across the entire translational spectrum. So we want to just emphasize that here. So in terms of our new and growing or fairly recent and new additions, we can map those into some very nice resources that are really good across the translational spectrum. So I'll go into more details here in a moment, but orofacial clefting data sets, public health, dental health data sets, and of course, temporal bone data on the way, and already in our collections. Working with HEAL initiative investigators to help them plan to contribute data to FaceBase in the near future. And then very clinical and clinically ready data sets around TMD and craniosynostosis. So we have both the Cranio Rate partners and the Human Craniosynostosis Atlas project as well. So just looking at what might be in, some example data sets here. In the orofacial clefting, some recent or some new additions here. As I mentioned, some of these could be very large. So we have what are probably some of the largest collections on orofacial cleft birth defects. So these OFC 1 and 2, courtesy of Mary Marizita and her collaborators at Pittsburgh and other institutes, have contributed data on thousands. So between these two, around 55,000 subjects. There's some overlap between those subjects, but really vast amounts of data collected from international sites that help identify the etiological factors of orofacial cleft birth defects. And then on the FORA and dental scores, we have public health data from underserved rural populations. We have dental score data, periodontal disease, cardiovascular genetic variants. As I mentioned, the TMD pilot work that's going on. So this is both gearing up to provide EMR data, which is in a fairly early stage, but we have some very good movement there, and we'll hear more about that, I believe, tomorrow. And we already have a collection of some highly structured clinical notes, some of probably the most well detailed and curated TMD clinical notes that you're going to find that are already that have already been submitted to FaceBase and will be available soon. And then we have new additions to the facial norms data. And as I mentioned, again, there's thousands of subjects with there's also clinical genetic diagnoses on these images as well. So it's a really rich collection relevant across the board. I want to highlight the NIDCD partnership. So this is the temporal bone database, aka what will be known as EarBase. And this is bringing really high resolution histology from the human temporal bone to FaceBase from 4 different sites that have been collecting this data in some cases for decades. So it's a really high value collection of data. Mass Eye and Ear, University of Minnesota, UCLA, Johns Hopkins. We did a lot of work. So just the theme of updates for the year 2. So we have done some really extensive database modeling of the patient case information on this. We've been working on enhancing our multi-tenant capabilities. We have a audiogram visualization prototype that we'll be expanding on in the coming year. We've done additional work in controlled access infrastructure to support this. And we'll hear a lot more about this, I think it is tomorrow, maybe. All right. And then our cranio-rate partnership. So this is a craniosynostosis clinical data federation. It is collecting and has collected craniosynostosis patient information. We are the downstream data repository as they collect things into their hub, their portal, and do their analytics on the data. We have an automated pipeline that will take that data to FaceBase. And in the past year, we've worked through public APIs, patient vocabulary alignment, data submission standards, both with cranio-rate as well as with the Human Craniosynostosis Atlas work to make sure these are aligned well, tested out our pipeline, and now we have the first 300 patients of that data with more to come. And so this also marks the first, I believe, the first clinical CT data, human CT data that we have in FaceBase, or among the first. And then the enamel-based partnership continues to produce data. So this is a consortium of amylogenesis and tooth enamel research. This brings stage-specific and single-cell gene expression data, enamel structure, composition, mechanisms, local proteome mapping, and related data. This has produced validated mouse models, an amelogenous primer, and standardized protocols. All of these are available on FaceBase. And we are receiving new data on this and will continue for the next few years at least. And then just a snapshot of all of the new data releases. We can map all of them to the various aspects of human health and human disease that these fall into. So whether it be tooth and enamel or cleft palate and craniofacial development, TMJ disorders, and then new data on spatial transcriptomics or single cell atlases. So I would encourage you to go and take a look at the FaceBase data browser, see the new data sets that are available. And then I just want to close out here, I think, with not whole presentation, but on the data that, as I mentioned, some of these have received new data. So those past slide was the 15 new ones. Then in addition, we have, of course, new data coming in, as I mentioned, on OFC 1 and 2, and also additional facial scans data. And then we have in the pipeline here, I believe these are fully curated but not released yet, but new highly curated cleft palate data, a new data set on microglossia. I think that's the first that we have on that condition. And then calvareal bone regeneration. And that's going to be the first rat model data that we have in FaceBase. All right, and then, all right, don't beat me up on this slide because I don't have exact numbers or exact details on these, but we wanted to give some sense of what the scale was of FaceBase, because as we're growing our collection, of course, we have the really huge behemoth sites of like DDGAP and UK Biobank that have some tens or maybe several tens of petabytes of data. And then all of us with maybe 10-ish or so. It's hard to get exact public details or authoritative on that, but I think that this is probably okay within orders of magnitude of the scale. So again, don't beat me up too much over that. But we wanted to just kind of point out that, when you think about FaceBases, we're getting a real critical mass of data, and it's in a concentrated area. We're not just everything across all types of anatomical regions and disease areas. It's a focused area. It's big. It's broad. It's very diverse in its own way. But we're starting to get a real critical mass. We're at about over 100 terabytes of data, well over 100 terabytes of data, on our way to a near-term projection of about 400 terabytes. And then over the next few years, especially with the NIDCD partnership, maybe looking at about a petabyte of data. And as we get more human subjects data, a lot of times it comes in where from different groups, a group we didn't know or expect comes in with 2, 3, or more terabytes of data. So there's really large collections amassing in FaceBase. All right. So one of the new resources that we've been focusing on in this past year, and this was in partnership with ClinGen to provide more clinically relevant data that they could use to enhance their own annotations for their own curators, is a spatial genomics atlas. And this work was really spearheaded and championed by Jifan Fang in our team, but really a full team effort. It required database model changes that Alejandro is working on, user study, user evaluation, and mock-ups that Chris is working on, and then user interface updates. But what we did is we built around a data set that Jifan had, plus she curated additional other data sets from different spatial genomics platforms. So we now have about 3 with a 4th platform on the way worth of different data sets that are building out the spatial genomics atlas in FaceBase. And currently this covers seepfish, busy merfish, and soon stereo seep in it as well. And I'll give a little canned demo of this as well, but it's very high resolution data on a cutting edge platform that we're now able to represent within FaceBase and have online visualization. So that leads into my next part of my next slide here on new platforms. I just want to highlight a few of the top things among really many that have gone in. So on the lines of this spatial genomics, so to do that, we added additional capabilities to our multi-channel viewer, and I'll just run the demo here. But what this does is that we can take these high resolution images, we can make them visible. You can do this all on the web. You go to this data set, and you can basically run this yourself, adjust hue and saturation. We can handle so the DAPI channel, as well as hundreds, even a few thousand. We're working up to being able to do the entire gene panel, 28 some thousand genes in one online viewer. But right now we're doing pretty well with hundreds and thousands. And you can go through that. If you noticed at the start of the presentation, I'll just back up. So this is really nice. You can zoom in to very high levels of detail. I'm going to just back up here. So if you notice, there were some annotations here. Now, you might have the NS, P, for palate, T for the tongue there. Each of these are linked on the left-hand side to a vocabulary term. So a controlled vocabulary term, you can drill down on that. You can find other data. You can look at what is the precise definition of that. You can then look for other data sets or other biosamples that are labeled with it. So you can get really, everything is just built to the core with fair data in mind. The next thing is we have a AI chatbot and semantic search capability. So you can use these new capabilities to search across FaceBase and support all sorts of interesting new modalities for finding data. I'm going to demo that later, so I'm going to just move. I'm going to keep you in suspense on that one for now. All right. And then we have an all new website refresh. So we wanted to enhance the website or the experience, especially for new users. So we're going to continue to serve our power users, but want to provide more of a gentle introduction for the new users. So we have kind of an updated look and feel here to the site. So we continue to have the nav bar and search box at the top of the page, so the power user could dive right into just looking for the data you want to find, and drill down to data sets. But for the newer user, provide more introductory material so that they kind of get a gentle on-ramp. So we'll have featured research that we will this will be updated throughout the year so we can feature new data sets. We have our refresh of our organism specific pages here, continue to have our mouse data summary. This is a full inventory. We have this also for zebrafish for capabilities there for any of our data. We're going to be adding disease specific research pages, and then we have our of course our solicitation, one of our favorite things, asking for new data, of course, and then a refresh of our news and publication pages as well. All right, and with that, okay, so one of the about that. So 2 minutes. Okay. So a lot of work has gone on in security and compliance. A huge area of effort has been around the new standard best practices for handling genomics data. We've made a lot of progress and expect to get to our first measure of compliance in a few months here. Effort has gone into our multi-tenant architecture to support these new sites. I just want to mention we have some important certifications and designations. We have CoreTrustSeal, of course. We're an NIH-designated CADR, Controlled Access Data Repo, NIH-designated Genomic Data Repository. We are approved for DMS, data sharing compliance, and then additional work has gone into our logging. And so, you want to find us. We usually show up at a number of these meetings: SCGDB, the Gordon Research Conference, the AADOCR, IADR. So those have all been part of our outreach in the past year, among others. And then just finally looking ahead, we will be having the launch of EarBase, the NIDCD temporal bone resource, on June 1st. We, as I mentioned, we already have cranial rate data. We will be expecting a lot more of that in the coming year. We'll be making more efforts into the AI and agentic tools. We have additional work planned around enhanced visualization, just general 3D visualization. And then we'll, of course, be engaged with the community and enhancing our DMS documentation. So again, thanks to our full team here for making all of this possible. And with that, I will conclude here. Thank you.
Okay, so do we have any questions? Are there any questions from our remote audience? Feel free to unmute yourself and just ask a question.
Hi, this is Stephanie Duda from Vanderbilt. I was curious if you're seeing submission of synthetic data sets to FaceBase, and if you have been thinking about how to handle those if they come in.
We've definitely been thinking about them. Alejandro, have we seen any yet?
We have one person from Colorado mentioned that he had the intention of submitting some craniosynostosis synthetic data.
That's right.
I think he was working in producing that.
Okay, yeah.
That's
so
the only that I remember.
Yeah, so if you heard Alejandro, yes, we have had one group has reached out about that. We're definitely thinking about that. I don't think we've gotten far enough to establish our sense for what the data format standard for that should be, but it's definitely on our minds. And also, I mean, we have another group that's from out of the US that has talked about synthetic data as well. So yeah.
I just wanted to add that we're having more inquiries regarding that of people wanting to submit synthetic data. Okay. So I think you guys start gearing up for that because you're going to be seeing more requests for people submitting and also asking for those data sets. And the rules about is it human controlled access to be determined because just because it's synthetic doesn't necessarily mean it can't be re-identified.
Right, absolutely. So as if you heard Nafa said so right. So especially synthetic data, if it's human based, it could still end up being controlled very possibly.
And the same question about controlled versus open data access would apply to data generated from, ex vivo human tissues.
Although I want to add that one of the reasons we're requesting synthetic data is so people don't have to do the whole control access thing, because it's mostly a lot of students want to use this. People want to use this to try things out before they actually then get the real data. So it's kind of like risk versus what are the trade-offs, and how restrictive we want to be.
In other settings, we've been looking at, so there are these basic questions of, if you're doing generative data, is there potential of leakage of control data through the model parameters? So we've been in other settings, particularly with fundus imaging, we've been looking at whether some of the models we're using will facilitate leakage. So it gets very technical and I don't understand most of it, but so we've also been kind of looking carefully at the controlled access in generative models and what you expose and don't expose.
Like what level of access? It might not necessarily be as high as human data, but maybe there might be not exactly open access.
Exactly. And so Stephanie, can you hear us talking?
Yes, I can.
Thank you. So Mike Fiolo, who is sitting in the audience, will immediately know what I'm about to refer to. That is the dbGaP practice of sharing summary level data through open access from human subject data still shared through controlled access. So I've been thinking about these new data forms, synthetic data, ex vivo data, all originally generated from human subjects, but might it be appropriate as deemed by, all the stakeholders, including data generators, data users, and regulatory experts, to share a summary level data from these quote unquote human data to allow potential data users to have a quick preview for potential study design.
Rafael, you have a question?
Yes, thank you. Can you hear me?
Yes.
Okay, yeah, I have a more generic question about how is the process to request the images? Because I think it was mentioned in the presentation that people can download the data and images to be analyzed offline. Is there a specific way or a mechanism for people to formally request that data to be used, and or some sort of agreement on what people can do with that image that is downloaded and the data, or even like connect with the lab who posted it to have some kind of a tracking mechanism?
Yeah, so all of any controlled access data that are that a researcher wants to use has to go through a formal data access request process, and that entails submitting documentation on their own IRB determination for being able to use that data, their own how they will keep that data secure, what their intended use for that data is, and it goes through the request goes to the NIDCD Data Access Committee that review and make a determination, and if approved, then the specific data or data sets are made available for either online or downloading by the requester.
Thank you so much. We've run out of time for the questions and answers, but thank you, Rob.